my name is Tivyaa Saravanan, and today our group will be presenting about forensic examination on body fluid, specifically into blood. Okay, and our group members are myself, Tivyaa Saravanan, Khoo Yong Tat, Usna, Siti, and Izyani. Okay, so these are the subtopic outline. Okay, so today we will discuss about two presumptive tests and three confirmatory tests. Okay, the presumptive tests are Kastle-Meyer test and luminol test. Okay, meanwhile the confirmatory tests are spectroscopic examination, microcrystal X-ray, and then microscopic examination of red blood cell and white blood cells. Okay, I give a short introduction to examination of blood in forensic science. Okay, so blood is a type of biological evidence which is commonly found at crime scenes. Okay, so blood stains are often found at various type of crime scenes such as hit and run, assault, child abuse, rape, robbery, and etc. Okay, it also uses for DNA extraction and establish the paternity test. Since bloods contain a valuable information, it is considered to be very important forensic tool. Analysis of different aspects of blood stain can contribute to clarify the circumstance under which some violent crime have been committed. Such a crucial information can point the criminal investigation in the right direction and help to solve the crime. In some cases, it also helps with legal determination of criminal offense, which can lead to more accurate and more appropriate punishment. Okay, there are various methods for blood identification, but today I will be presenting about a chemical examination, which is a phenolphthalein test, also known as a Kastle-Meyer test. Okay, so now let me share about you the history of this test. Okay, so the emergence of the phenolphthalein test was from the discovery of a technique for the detection of plant peroxidase by Joseph H. Kastle and Oliver March in 1901. Okay, later in 1903, Meyer used the test for the detection of blood and pus fluids. That's the reason why it called as the Kastle-Meyer test. Okay, let me explain to you about the principle of this test. Okay, so the principle of Kastle-Meyer test is based on the resultant reaction in which phenolphthalein, which is a colorless compound, catalyzed by heme with hydrogen peroxide as the oxidant. Okay, so the resultant reaction consists of reduced phenolphthalein is an alkaline solution to produce phenolphthalein of a bright pink color. Okay, so the pink color phenolphthalein is carried out only in alkaline condition. So in the following slide, we will go through how to perform a Kastle-Meyer test. So but before that, we will just listen what are the material we needed for this test. Okay, so we need Kastle-Meyer solution, 70% ethanol, distilled water, 3% of hydrogen oxide, cotton swab, pipette, and dry blood. Okay, now let me explain to you how to perform a Kastle-Meyer test. Okay, so first of all, we need to moist the cotton swab. Okay, and then we need to touch the suspected blood with the cotton swab. Okay, and the important point is swabbing and rubbing is unnecessary because of the high sensitivity of the test. And the third step is optional. Okay, so you may apply a drop of 70% ethanol to the swab. Okay, because this will help the alcohol to breaks and opens the blood cell and exposes the hemoglobin, which will help in improving the test sensitivity. Okay, so the fourth step will be apply a drop of Kastle-Meyer solution to the cotton swab. Okay, initially the solution is colorless or pale yellow. Okay, it should not discolor the cotton swab. If the drops are pink, the solution is old, which is oxidized and will not work. If the swab immediately change color upon Kastle-Meyer solution application, it either means the solution is old or else there is an oxidizing compound present. In either case, retest using a fresh Kastle-Meyer solution. Okay, and the fifth step will be wait for a few seconds. Okay, now apply 1 or 2 drops of 30% hydrogen peroxide. Okay, and immediate color change to pink is a positive test for blood, and eventual color change occur whether the blood is present. Let me explain to you observation of this test. Okay, the result of this test. If there is the immediate changes of color seen, which is from colorless to pink, it's considered as a positive test result. Okay, and if there is no changes in color, it's considered as negative test result. Okay, now let me explain to you about the false positive and false negative test result. Okay, so Kaplan-Meier test is a presumptive test. Okay, so which means a negative results rule out the presence of blood, while a positive test indicates a sample is probably blood. Okay, however, a positive test results really only indicate the presence of hemoglobin, which comes from red blood cells in a mammal and other animals. Okay, but bacteria and even plants can produce hemoglobin under the right circumstances. Now let me explain to you about the advantage and disadvantage of this Kaplan-Meier test. Okay, so the advantage will be it is known to be one of the specific tests for the detection of blood with a less false positive result. Okay, other than that, the reagent are also known to be a non-carcinogenic. Okay, and it also develop a positive color reaction of up to dilution level of 1 to 1 million. Okay, in solution. So the disadvantage of this test is it is less sensitivity than the benzidine test. Okay, other than that, it also hinders the DNA analysis by reduce the amount of recoverable DNA. Okay, other than that, it also less sensitivity than other comparable screening tests for blood. Okay, now let me explain to you about the other limitation of this test. Okay, so this test is sensitive enough that it detects the blood diluted down ratio is 1 to 10. Okay, this is roughly 1 drop of blood in 10,000 drop of water. Okay, while this pretty dilute, trace quantities of blood might not cause the color changes. Okay, the original test uses a swab dam with water. Okay, the treating the swab with ethanol lyses the cells and improve the sensitivity of the test. Okay, but theoretically the test is non-destructive, which meaning you can use the blood on the swab for further analysis. Okay, however, the sodium hydroxide solution actually does degrade the DNA. Okay, the usual practice is taking separate blood sample for analysis if possible. Okay,
and now we move to the second presumptive test of analyzing the blood by using luminol solutions. Let's say if we have a murder case, a murderer can dispose of the victim's body and mop up the pools of blood, but we have to know that without some heavy-duty cleaning chemicals, some evidence will remain. So the tiny particles of blood will cling to most surfaces for years and years without anyone ever knowing that the blood is there. Hence, we use the luminol test. So the basic idea of luminol is to reveal these traces with a light-producing chemical reaction between several chemicals and hemoglobin, which is an oxygen-carrying protein in the blood. Investigator will spray to the suspicious area, turn off all the lights, and block all the windows and look for a bluish-green light. If there is any blood traces in the area, then the luminol solution will glow, indicating the positive reaction of the luminol solution. And now we will talk about the methodology and about mechanism behind how luminol works. The central chemical in this reaction is luminol, a powdery compound made up of nitrogen, hydrogen, oxygen, and carbon. Criminalists will mix the luminol powder with a liquid containing hydrogen peroxide, which is a hydroxide, and other chemicals, and pour the liquid into the spray bottles. So the hydrogen peroxide and the luminol are actually the principal player in the chemical reaction, but in order to produce a strong glow, they need a catalyst to accelerate the process. So the mixture is actually detecting the presence of such a catalyst. In this case, is the ion in hemoglobin. To perform a luminol test, the criminalists simply spray the mixture wherever they think the blood might be. If hemoglobin and the luminol mixture come in contact, the ion in the hemoglobin accelerates a reaction between the hydrogen peroxide and the luminol. In this oxidation reaction, the luminol loses nitrogen and hydrogen atoms and gain oxygen atom, resulting in a compound called 3-amino phthalate. The reactions leave the 3-amino phthalate in an energized state. The electrons in the oxygen atoms are boosted to higher orbitals, so the electron quickly fall back to a lower energy level, emitting the extra energy as light photon. With ion accelerating the process, the light is bright enough to see in a dark room. Next part, we will discuss the practicality of luminol tests. Firstly, about the pH. Considering that pH values between 10 and 13 enable optimal conditions to luminol reaction, the medium pH must remain alkaline in order to increase the test efficiency. Prior experiments have demonstrated that pH 11.5 is ideal to identify blood spots. The next aspect is the intensity of the light. Depends on the nature and concentration of the catalyst used. It may present, and this effect can be photographed and lasting approximately 1 minute. Moreover, the test requires the application of luminol solution in the blood stain, whether visible or not to the naked eye, just to prove the results. Next, we will discuss about the accuracy of luminol tests. The major benefits of performing a luminol test rather than one of the colorimetric tests is that one can search large area very rapidly, and the chemical uses doesn't interfere with any other of presumptive tests that may be performed following luminol testing. If the result of presumptive test is negative, then the analysis is terminated. However, if the result is positive, then a more definitive confirmatory test is performed. Second, luminol shows higher sensitivity, making feasible the detection in nanogram scale. For this reason, depending on the sample concentration, it is the best alternative for the analysis. Thirdly, luminol solution presents also the advantage of maintaining the integrity of the genetic materials after the analysis. And lastly, a successive wash with hypochlorite solution promotes the accumulation of this ion, which increases the intensity of light emission by luminol. And now we will talk about these advantages of luminol. So the result of luminol test may be altered by a broad range of interfering compounds, and hence the false positive results. The reasons for this is that there are a number of substances that can produce false positive observations, including bleach, plant peroxidase, chemical oxidants such as potassium permanganate, copper, lead, zinc, iron, and even cobalt. The second disadvantage is the clearance of the blood stain upon washing with hypochlorite solutions will remove more hemoglobin compared to the water alone. As the hemoglobin concentration decreases, the emission of light by luminol also will be reduced. Not to mention, the medium where the test is carried out is composed of sodium hydroxide, soda, hydrogen peroxide, and sodium perborate. So these components are considered irritating to the eyes and mucus, thus the use of personal protective equipment is essential. Not to mention, federal departments doesn't control the sale of this product, and only a few studies about its toxicity are available in the literature. So in conclusion, based on the result that we have discussed just now regarding two presumptive tests, the luminol test is clearly the most sensitive blood detection technique commonly used by forensic investigators. This is because the interesting finding was that the sensitivity of the Kastle-Meyer test decreased considerably when applied to cotton swabs of the blood stains. However, the sensitivity of the luminol test was found to be 1 to 5 millions in ratio for both the blood stain clothes and hemoglobin solutions. Furthermore, luminol tests can maintain genetic material in the blood, while Kastle-Meyer will hinder the DNA analysis due to the strong chemical reaction. And therefore, the luminol test is by far the most sensitive technique and easy to perform, particularly when application over a large area is required.
Hi guys, my name is Khoo Yong Tat. Next, I will explain about the comparison between the presumptive test and confirmatory test. First of all, the presumptive test has narrow possibilities and it helps decide which test to do next, while confirmatory tests conclusively identify and confirm the blood. Next, presumptive test is high in sensitivity, while confirmatory test is high in specificity. After that, presumptive test can be carried out at the crime scene, but confirmatory test must be done in laboratory. This is because presumptive test takes shorter times than confirmatory test. Moreover, confirmatory test requires more specific additional equipment. Last but not least, confirmatory test has smaller risk of false positive compared to presumptive test. Next, we will explain further about the confirmatory test of blood. So first of all, microscopy examination. We will have two types of microscopy examination, which are microscopy examination of instant red blood cells and white blood cells. So first of all, microscopy examination of instant red blood cells is an examination technique used to observe the structure of red blood cells using the microscope. and is only used in fresh or clotted blood samples. The procedure. Firstly, the stent sample was taken and is placed inside a petri dish. Second, with a dropper, 2 to 3 drops of white blood fluid were added, and you should make sure stent clot fully stopped. Okay? Next, wait for at least 6 hours, and the stent extracted was removed and placed on a glass slide. Lastly, the glass slide was covered with a cover slip and it was observed under a microscope. The observation was recorded. So here's a note. White blood fluid is a mixture of sodium chloride, mercury chloride, and distilled water, or we use the normal saline. If the blood sample is in liquid form, you can directly observe under the microscope. But if the stent is unclogged, soak the clogged sample in normal saline or white blood fluid for at least 6 hours. However, the recommended time to be soaked is 12 hours. So, here are the comparison of structure among the human red blood cells, mammalian red blood cells, and non-mammalian red blood cells. So, first of all, the structure. The structure of human red blood cells are circular, biconcave, and non-nucleated, while the mammalian red blood cells are similar to human red blood cells. However, the non-mammalian red blood cells are oval, biconvex, and nucleated. Next, the size. The human red blood cells have average diameter of 7 micrometer and 1.7 to 2.2 micrometer in thickness, while the mammalian red blood cells have 6 to 7 micrometer in diameter. However, the size actually depends on the type of mammalian blood. Next, the non-mammalian red blood cells have no definite size. It actually depends on the type of non-mammalian blood. So we can conclude that only the human red blood cells have the fixed diameter and the thickness. So here are the assumptions for the structure of the human red blood cells, which are the newborns and the patient with plant vision or the late progeny have nucleated red blood cells. Next, the mammalian red blood cells also have some exceptions in the shape and also the presence of nucleus. So the camels have the elliptical shape and the lemurs have nucleus in the red blood cells. Next, I would like to discuss further about the microscopic examination of white blood cells. It is an examination technique used to count the division bodies in polymorph cells. It is used to identify the genetic sex of a person. The presence of division bodies in polymorph cells indicates that the person is phenotypically female, while the absence of division bodies indicate that the person is phenotypically male. The procedure. Firstly, the stent sample is taken and is placed inside a petri dish. With a dropper, 2 to 3 drops of Leishman stent were added onto the blood stent. Lastly, the glass slide was covered with a cover slip and is observed under a microscope, and the observations are recorded. Here's a note. Leishman stent is used as a blood smear agent for differentiating and identifying the white blood cells and mammalian parasites. So in the picture, the arrow pointed the presence of division body in white blood cells. Therefore, the person is phenotypically female. Next, here are the features of both microscopic examination of red blood cells and white blood cells. First of all, the validity. The light microscope and electron microscope are valid for the both microscopic examination. Next, accuracy. For microscopic examination of red blood cells, it's difficult to differentiate the human blood with other mammalian blood, but it's still possible. As we know, the human red blood cells have the fixed diameter and the fixed thickness compared to other mammalian blood. So we can differentiate based on this point. And then, for the microscope examination of white blood cells, the presence of division bodies only indicate the sex of human. So they have some advantage. So both of them are high magnification and high resolution, and they are easy to use. However, they still have some disadvantage. So firstly, they are only used for the fresh or clotted blood samples. Next, the electron microscopes are expensive and big in size, while the lab microscope cannot operate in the darkness.
So next, I will present about microcrystal assay test. So microcrystal assay test or microcrystal test can be defined as a chemical test that result in the distinctive morphological development of microcrystal crystal as a sign of blood conformations. So this test is commonly used and widely used by scientists as it has a lot of advantage. So one of the advantage of this test is it has a really simple and rapid procedure. Although this test is considered as a good confirmatory test, this test still has a disadvantage. So one of the disadvantage of this test is it is less sensitive, as we cannot define whether the blood sample is from human or non-human. Other than that, this test holds a principle which is a chemical reactions between a substance and a reagent that yields a crystal formation that can be observed under a microscope. So different microcrystal assay tests produce different shape of crystal, but almost react the same with hexavalent. So these tests have 3 types of tests, but in the presentations, I will only discuss about 2 tests, which are Takayama test and attachment test. So the first test that I will discuss is attachment test. The principle of this test is based on direction of the heme part of blood with sodium chloride and glycolic acid to form a brown colored rhombic crystal. So these rhombic shaped brown crystal are a sign of the formation of hematine chloride crystal. So this slide is about the procedure of attachment test. So attachment test contains 5 simple steps. So the first step is that a drop of blood is dropped on the slide, and then a few drops of paper reagent is added. So the reagent we use in attachment test is sodium chloride and glycolic acid. Thirdly, the slide is then covered with a slip cover, and then the slide is heated for a few seconds until the first bubble is formed. So lastly, the slide is observed under a microscope. So for this slide, I will discuss about the positive version of the test. So for attachment test, the positive result is when the last product for this test is the formation of rhombic crystal that are brown in color and arranged in a singly or in cluster. Next, the reagent used in attachment test is sodium chloride and glycolic acid. The advantage of the attachment test. So the advantage of this test include the test is easy to perform as this test is really simple. Secondly, this test is highly specific to blood. And lastly, only 2 sets of reagents are required. Meanwhile, the disadvantage of the test is it is not reliable for all blood stands. So the second test is Takayama test. So the principle of the test is based on the formation of hemochromogen with a reaction with pyridines. Next is the method for Takayama test. So the first step is that the dried blood stain sample is scrubbed and is put it over a glass slide. And secondly, a few drops of repair reagent is added. So the reagent we use in this test is saturated glucose solutions, sodium hydroxide 10%, and pyridine. And thirdly, the slide is then covered with a slip cover. And lastly, the slide will be observed under a microscope. Next is the positive reaction of this test. So for Takayama test, the positive result is when the last product for the test is the formation of feather crystal in pink and is arranged in cluster or sheaf. So the reagent we use in this test is saturated glucose solution, sodium hydroxide 10%, and pyridine. Next is the advantages and disadvantages of Takayama test. So for the advantages of Takayama test, first is it is best for all blood stains. Secondly, just a small amount of stain sample is needed. Thirdly, we doesn't use any corrosive reagents. And last but not least is no heating process is required. Meanwhile, the disadvantages of using this test is that this test require a longer time to perform compared to attachment test. So for this test, the examiner has to wait for at least 30 minutes to get an accurate result. So next, I will discuss about the reliability of attachment test and Takayama test. So between Takayama test and attachment test, Takayama test is more reliable than attachment test. This is because attachment test cannot use for all blood stain. However, Takayama test is best used for all blood stains. Not only that, Takayama procedure is much more simpler than attachment test, as Takayama test doesn't use any corrosive reagent and doesn't involve heating process. So next is the discrimination power of Takayama test and attachment test. So among the confirmatory tests, the Takayama test is more promising than attachment test, and it has been proven by the researcher from Department of DNA or Biology Division Center for Forensic Science in India. So the researchers and scientists at this department has conducted a study to identify the most sensitive confirmatory tests of blood among tacrolimus test and attachment test. So in this study, the confirmatory tests were performed on washed blood stained fabric, and the fresh blood was poured uniformly on the cotton fabric to make a blood stain. And those fabrics was kept for drying for 10 days. And after the cloth is dried, it was washed using a various washing step. So firstly, A1, the stained cloth was dipped in water for 2 hours. Secondly, B1, the stained cloth was washed with water for 60 minutes. Last but not least, C1, the stained cloth was dipped in detergent water for 2 hours. Then, the washed blood stained fabric were tested for the presence of blood to know the detection efficiency and sensitivity of different methods. So, according to the result, when the A1 stained fabric is tested with Takayama test, it gives a strong positive result. Meanwhile, for attachment test, it just gives a mild positive result. When B1 is tested with Takayama test, it gave mild positive result and weak positive result for attachment test. And lastly, for C1, it gave weak positive result for Takayama test and negative result for attachment test. So therefore, for washed blood stain on fabric, the Takayama test is the most suitable confirmatory test. So we can see that Takayama test has a higher discrimination power compared to attachment test. So in conclusion, I can conclude that Takayama test is more accurate than attachment test, as Takayama test has a higher discrimination power and more reliable compared to attachment test.
My name is Izyani Binti Zulkifli, and I will be presenting spectroscopic blood examinations. Spectroscopy technique for blood stain examination is considered to be one of the most reliable confirmatory tests in blood stain pattern analysis because it studied various functions, especially in detecting blood, or to be precise, hemoglobin and its derivatives. It distinguishes human, non-human blood with any other bodily fluids and measuring the age of blood stains. It is also a non-destructive instrument. The simple procedure of this method starts with filling 0.1 mg blood sample in a test tube, and the sample will be diluted with normal saline. Then pour the small amount of sample into cuvette before placing it into the spectrometer. What to observe in this examination is the dark absorption bands that differ based on hemoglobin and its derivatives. The following absorption spectra shows the Fraunhofer lines that can be found in all of the hemoglobin and its derivatives. These are a set of spectral lines extended to the full spectrum that are used as the metric to categorize the different absorbed spectra. The wavelengths in the spectra are labeled A, B, C, D, E, and F, G. To observe, the oxyhemoglobin in the spectra shows two distinct bands in yellow between Fraunhofer lines D and E. One band is near D and half the breadth of the other and more defined. Next, in reduced hemoglobin, the band is the broad band that lies between D and E, while the carboxyhemoglobin, it is similar to oxyhemoglobin and remains unchanged. Even oxyhemoglobin get reduced by adding ammonium sulfide. In methemoglobin, two similar bands are of oxyhemoglobin with two extra bands. The third dark band lies in the red region between the Fraunhofer line C and D, and the fourth line lies between E and F, which is more indistinct. Whilst in acid hematine shows a sharp band between C and D with a broad band between D and F but not well defined. Next, in alkaline hematine, one band can be seen lies between C and D, and in hemochromogen, one dense narrow band lies midway between D and E, and the second broad and pale band lies over E. Cyan hemochromogen spectra recorded to have similar bands to hemochromogen but slightly wider. Next, for acid hematoporphyrin, a sharp broad and dark band seen to lie between D and E. And lastly, for alkaline hematoporphyrin, one band lies between E and F is the broadest. Two bands are in between D and E, while the fourth band between C and D. Next, I will be explaining about various spectroscopy techniques with its studies, functions, advantages, and limitations. In Raman spectroscopy, it holds the most promise for future use in the field of bloodstain pattern analysis. This method is highly sensitive and non-destructive to the sample. Raman spectroscopy is superior to the other techniques for the identification of blood in the presence of contaminants. This method can also be used with portable devices, allowing for on-site analysis, which increase the overall efficiency of evidence examination. Research has demonstrated that Raman spectroscopy has the ability to differentiate between fresh blood and old blood, but cannot determine the specific age of bloodstains. Therefore, this application of Raman spectroscopy should be considered preliminary, and further research is required to enhance the accuracy of bloodstain estimations. Studies should also focus on applying this technique to extensively aged bloodstains, since bloodstain pattern analysis are not always assessing crime scenes that are less than 1 week old. Nevertheless, recent studies have attempted to determine the gender, ethnicity, and even general age, for example, adult or infant of blood donors. But Raman has not yet proven to be a robust technique for this analysis. Even analysis of the control groups yield only moderate to low accuracy specificity rates. A common issue with these recent studies is that small representative sample sizes were used, and it is unclear if any of the detected difference can be considered true results. Raman spectroscopy also only able to estimate up to 1 week old bloodstains. IR spectroscopy appears to be promising technique for bloodstain pattern analysis, especially since it has been studied extensively and is well understood. It is also displayed overall moderate to high accuracy, depending on what substrate was involved. In addition, it has the ability to be coupled with handheld devices. Portable IR-based methods have been reported as being very easy to use, which is extremely advantageous in instances where the investigative personnel lack a scientific background. One study used near IR spectroscopy to differentiate blood from non-blood substances on dark and colored cotton fabrics for up to 1 month after deposition. This research showed promising results in that the technique was able to identify blood and even estimate its age, with an error rate of 8.9%. Compared to other studies, this error rate is relatively small. However, only cotton fabrics were assessed, meaning that this method is limited in its forensic applicability. Overall, some validation is still required for specific bloodstain analysis, but this technique performs well for the differentiation of human from other species blood and blood-like substances, and can be recommended as a preliminary testing method. ATR-FTIR spectroscopy is a study under IR spectroscopy. This related study showed the most promising potential as it studies the estimation of bloodstain ages in indoor and outdoor settings for up to 107 days. It was determined that this technique was successful in the age estimation of indoor and outdoor bloodstains between 7 to 85 days with high accuracy of R-squared 0.94 for indoor stains and 0.96 for outdoor stains. This method was also able to differentiate fresh bloodstains of less than 1 day old and older bloodstains of longer than 1 day old. Aside from that, another study demonstrated that FTIR-ATR spectroscopy has the ability to distinguish between antemortem and postmortem blood stainness, even on dark surfaces such as black dyed polyester cloth and blue. This study claimed to be highly accurate with the ability to detect and differentiate blood 86 to 95% of the time. However, this considers the weighted average of the results. On the other hand, recent research into reflectance spectroscopy has searched. Reflectance spectroscopy has potential for specific analysis of bloodstains on smooth and raw surfaces, and is adequate for detecting and distinguishing blood. Therefore, this method may be useful as a presumptive test. Bloodstain age estimations were achieved with only mediocre accuracy using reflectance spectroscopy, and thus this method cannot be recommended for such analysis. However, the specificity of reflectance spectroscopy has been demonstrated to be quite high and may be useful in cases where blood and non-blood substances are present. Distinct, specular, and diffuse reflectance spectroscopies are not typically pairable with portable devices, meaning that on-site analysis are not supported. Also, it has been established that this technique can be impacted by factors that may affect light scattering, such as substrate interference. This has resulted in varying error rates across a number of studies, which should be taken into consideration before deciding to use in the field. Proceeding to the next technique, which are NMR and EPR spectroscopy. These technologies have great potential for the development of specific blood profiles that could be used for the identification and differentiation of blood. But unfortunately, these techniques are complex and not supported by portable devices, meaning that specialists and laboratory analysis are required. EPR spectroscopy has been used to estimate the age of bloodstains on filter paper and goes over 775 days with consideration of the potential effects of various environmental factors on analysis. Strong correlations were determined between EPR signal intensity and time since disposition of up to 432 days, while the potential to examine bloodstains over a year is intriguing and may seem significant to forensic analysis. This technique displayed error rates of up to 25%, indicating that as bloodstains age, the accuracy of this method decreases to an extent that its applicability is diminished. Furthermore, it has been previously established that hemoglobin and other blood components undergo chemical changes as bloodstains dry, and as these alterations are dependent on environmental factors such as temperature. Nonetheless, these techniques are more expensive to operate and maintain, and sample preparation is needed before analysis can be conducted. This leads to destruction of the sample, meaning that these techniques are not favorable if only a small amount of evidence is available. Thus, very few bloodstain studies have been performed using NMR and EPR. This means that NMR and EPR sample may not be appropriate for downstream analysis such as DNA analysis. These techniques are highly sensitive and accurate, but because of the limitation associated with this method, not many studies has been performed. Absorbance techniques such as UV visible spectroscopy are easily combined with portable devices, allowing for on-scene analysis, but they require preparation of the sample prior to examination, which is not favorable in situations where only a single small sample is available for testing. The sensitivity of these techniques mean that very little sample is required in order to perform analysis. On the other hand, atomic force spectroscopy is another method that can be made portable for rapid on-site acquisition of results. This technique is also advantageous in that no sample preparation is required prior to analysis, thus decreasing the evidence examination time even further. However, the limitation for absorbance technique lies on its method that lacks specificity for differentiating human blood from animal blood and other bodily fluids. and therefore should only be used as a presumptive test for the potential presence of or absence of blood. Whilst for atomic force spectroscopy, it can be influenced by many different factors such as presence of contaminants and exposure of the evidence to the environment. Due to these influencing factors, the accuracy rates of atomic spectroscopy has not yet been determined. Specifically with regards to bloodstain age estimation, many validation studies would be required before this technique could be considered for use in forensic context. All in all, many of these studies discuss shared similar limitations and the need for further validation and optimization. Through research, these techniques are continually being refined. While certain research endeavors aim to help in the investigation of cold cases or old crime scenes, there is no one technique that can be used to produce accurate and reliable results that will withstand the scrutiny of the courts for all these topics. Although by comparing these techniques, Raman spectroscopy suggests the better technique to solve many contexts of varied cases, still more focus should be applied to the effects of environmental factors on bloodstain analysis with spectroscopic methods. The current research endeavors are often limited by sample size or only focus on limited number of substrate types. Future research should encompass a greater variety of substrates including plastic, which is a very common material in society. In summary, spectroscopic techniques have great potential to be highly useful tools in bloodstain pattern analysis, but much more is still required before their limitations are fully understood, and their use can be recommended as standard operating procedures by practitioners in the field. To compare between the confirmatory tests discussed in this video, which are microscopic examination of red blood cells and white blood cells, microcrystal acid tests, or spectroscopy blood examinations, I think spectroscopy technique suggests the most promising potential. While there is no one among specific spectroscopy technique that promise of accuracy nor reliability of the outcome, more understanding of its limitations to be explored or validations of studies to seek, these techniques are continually being refined with studies of various cases to produce more potentially qualified results. I think that's all from our group. Thank you.